Okay, so chapter 3 is going to go over cells. Cells are the smallest living units, and we have what's referred to as a cell theory. A cell is the structural and functional unit of life, and the human body is going to be made up of over 200 different cell types. Uh, how well the entire organism functions depends on the individual and combined activities of all of its cells. The structure and function are complementary, meaning that the structure of a particular cell, and you're going to see human cells have many different structures, really dictates its overall function. Biochemical functions of cells are dictated by the shape of the cell and its specific subcellular structures. The continuity of life has cellular basis, and cells can arise only from other pre-existing cells. So there are over 250 different types of human cells. So there's a lot of cell diversity. One thing that I do want to point out is that even though we have over 250 different types of human cells, we do all those cells will still have the 46 chromosomes, or aka the 23 pairs of chromosomes. That does not change. These cells will differ in size, shape, and subcellular components, meaning organelles, and these differences lead to the differences in their functions. And here's just some diversity. We can see erythrocytes, which are red blood cells, fibroblasts, epithelial cells. This is what we typically find in structures like the skin, muscle cells, fat cell, macrophage, which is a white blood cell, nerve cell. You can see this is very long. This can actually be several feet long. And then a sperm cell. Sperm cell is actually the only cell in the human body that has a flagella. So a generalized cell, all cells have some common structures and functions. Human cells have three basic parts. They're going to have a plasma membrane, which is a flexible outer boundary made up of a phospholipid bilayer. They will have a cytoplasm, which is intracellular fluid containing organelles. And then you will have a nucleus. This is a DNA containing control center. And here we can see a generalized structure of the cell. I believe you guys are actually labeling a picture very similar to this for your either lecture assignment or lab assignment, I forget, but it's one, I think it's your lab assignment. So this will be make sure you come back to this slide for your lab assignment there. We're going to go over all these structures. So extracellular materials, these are substances found outside cells. And classes of extracellular materials include extracellular fluids, such as body fluids, interstitial fluid, these are cells are submerged or bathed in this fluid. You have blood plasma, this is the fluid of blood where you're going to have red blood cells, white blood cells, and platelets suspended in. Cerebral spinal fluid, this is the fluid surrounding the nervous system organs. Cellular secretions, such as saliva and mucus. And extracellular matrix, this is substance that acts as a glue to hold cells together. So the plasma membrane, this acts as an active barrier separating intracellular fluid from extracellular fluid. Plays a dynamic role in cellular activity by controlling what enters and what leaves the cell. It's also referred to as the cell membrane. Animal cells do not have cell walls, so we will not be discussing that structure here. That is a unique feature with animal cells. So the structure of the plasma membrane consists of membrane lipids that form a flexible lipid bilayer. You'll also have specialized membrane proteins that float through this fluid membrane, resulting in constantly changing patterns. It's referred to as a fluid mosaic model made up of many pieces or pattern. You have surface sugars, which will form what's called a glycocalyx. And then you'll have membrane structures, which will help to hold the cells together through cell junctions. And looking at a typical plasma membrane, I like this figure a lot because it really defines the different components. You can see there's phospholipids and why they're important. Cholesterol, why that's important, it really adds rigidity to the membrane. The various types of proteins, and then the carbohydrates and what their functions are. So I would definitely take a minute and look at this slide and look at the key components there. Okay. So for example, looking at the functions of plasma membrane proteins, functions of the plasma membrane, it acts as a physical barrier. It's selectively permeable. What that means is it can restrict what can and cannot enter or exit the plasma membrane. It's involved in communication with the outside world, as well as cell recognition. So the lipid bilayer is made up of pretty much 75% phospholipids, which consist of two parts: phosphate heads, which are polar, and they are hydrophilic, which means they like water, and fatty acid tails, which are nonpolar, which means they are hydrophobic and they do not like water. So that's about 75% of the plasma membrane. 5% of glycolipids. These are lipids with sugar groups on the outer membrane surface. And then 20% is cholesterol. This increases membrane stability. Cholesterol is essential for the plasma membrane. Here's a typical structure of a phospholipid. Again, this part is water loving. This part does not like water. And what you can see is that the outer surface here is okay. Most likely it's going to be composed of water or a watery substance, but that's okay because you can see this part is okay with water, then does not like water, so this sets up the barrier. And the inside of the cell typically is going to be a cytosol, which is mostly composed of water as well. So that's why you see this bilayer with these two layers sort of setting up that barrier. So membrane proteins will allow cell communication with the environment. They make up about half the mass of plasma membrane. Most have specialized membrane functions. Some will float freely, and some are tethered to intracellular structures. And there are two types: integral proteins and peripheral proteins. Integral proteins are firmly inserted into the membrane. Most are transmembrane proteins. They have hydrophobic and hydrophilic regions. The hydrophobic areas interact with lipid tails. The hydrophilic areas interact with water. The function as transport proteins, which means these are going to be channels and carriers. Integral proteins can also be enzymes or receptors. Peripheral proteins, these are loosely attached to integral proteins. And they include filaments on the intracellular surface, which is used for plasma membrane support. It will function as enzymes, motor proteins for shape change during cell division and muscle contraction, as well as cell-to-cell connections. Membrane proteins can perform transport, which is a protein that spans the membrane, and it may provide a hydrophilic channel across that membrane that is selective for a particular solid, meaning that these are very specific for what they allow to transport through. Some transport proteins hydrolyze ATP as an energy source to actively pump substances across the membrane. Receptors for signal transduction. A membrane protein exposed to the outside of the cell may have a binding site that fits the shape of a specific chemical messenger, such as a hormone. When bound, the chemical messenger may cause a change in shape in the protein that initiates a chain of chemical reactions in the cell. Enzymatic activity. This is a membrane protein, may be an enzyme, with its active site exposed to substances in an adjacent solution. A team of several enzymes in a membrane may catalyze sequential steps of metabolic pathway, as indicated left to right here. Cell-to-cell recognition, some glycoproteins. These are proteins that are bonded to make short chains of sugars, which help to make up the glycocalyx and will serve as identification tags that are specifically recognized by other cells. And then you have the attachment of the cytoskeleton and extracellular matrix. Elements of the cytoskeleton and the extracellular matrix may anchor to membrane proteins. This helps maintain cell shape, fixes the location of certain membrane proteins, and plays a role in cell movement. Cell-to-cell joining. Membrane proteins of adjacent cells may be hooked together in various kinds of intracellular junctions. Some membrane proteins of this group provide temporary binding sites that guide the cell migration and other cell-to-cell interactions. So the glycocalyx, this consists of sugars or carbohydrates sticking out of the cell surface. Some sugars are attached to lipids, glycolipids, and some to proteins, which are glycoproteins. Every cell type has a different pattern of the sugar coating. This functions as specific biological markers for cell-to-cell recognition and allows the immune system to recognize self versus non-self. Cell junctions, some cells are free, not bound by any other cells. Most cells are bound together to form tissues and organs. Three ways cells can be bound to each other are tight junctions, desmosomes, and gap junctions. Tight junctions, these are integral proteins on adjacent cells which fuse to form an impermeable junction that encircles the whole cell. It prevents fluids and most molecules from moving in between the cells. So where might this type of connection be useful in the body? And the answer would be where you want to set up a particular barrier. Here you can see the tight junction. So you're setting up pretty much a very, very distinctive barrier here where things are not going to be able to pass through the cells. The desmosomes, these rivet-like cell junctions formed when linker proteins of neighboring cells interlock like the teeth of a zipper. Linker protein is anchored to the cell through a thickened button-like areas on the inside of the plasma membrane called plaques. Keratin filaments connect plaques intercellularly for added anchoring strength. Desmosomes allow give between the cells, reducing the possibility of tearing under certain tension, and that's what these structures are here. So you can see how they sort of interlock with each other. Gap junctions, these are transmembrane proteins, which will form tunnels that allow small molecules to pass from cell to cell. It's used to spread ions, simple sugars, or other small molecules between the cells. It allows for electrical signals to be passed quickly from one cell to the next, and it's used in cardiac and smooth muscle cells. And you can see these little gap junctions, these little channels between cells. Many substances constantly move across the plasma membrane. Some molecules pass through easily, some do not. The plasma membrane is selectively permeable, allowing only certain molecules to cross. You have two essential ways substances cross the plasma membrane. Passive transport, which is no energy is required, and then active transport, where energy or ATP is required. So passive transport requires no energy input. There are three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. All types involve diffusion, natural movement of molecules from areas of high concentration to areas of low concentration. It is also referred to as moving down a concentration gradient. All molecules have random high-speed movement due to their intrinsic kinetic energy. Movement results in the collisions between molecules. Molecules in higher concentration areas collide more, resulting in molecules being scattered to lower concentration areas. And here you can see an example of diffusion. The speed of diffusion is influenced by three factors: concentration. The greater the difference in concentration between the two areas, the faster the diffusion occurs. Molecular size, smaller molecules diffuse faster, and temperature, higher temps increase kinetic energy, which results in faster diffusion. Equilibrium is reached when there is no net movement of molecules in one direction only. Molecules have a natural drive to diffuse down concentration gradients that exist between extracellular and intracellular areas. Nonpolar hydrophobic lipid core of protein membranes stop diffusion and create concentration gradients by acting as selectively permeable barriers. It is also called differentially permeable barrier. Molecules that are able to passively diffuse through the membrane include lipid-soluble and nonpolar substances, very small molecules that can pass through the membrane or membrane channels. These are referred to as simple diffusion. Larger non-lipid soluble or polar molecules can cross membrane, but only with assistance of carrier molecules, which is referred to as facilitated diffusion. Osmosis is a special name for movement of a solvent, usually water, not molecules. Simple diffusion is when you have nonpolar lipid-soluble substances diffused directly through the phospholipid bilayer. Examples include oxygen, carbon dioxide, steroid hormones, fatty acids. Small amounts of very small polar substances such as water can even pass through. So here you can see lipid-soluble substances passing through by simple diffusion. Facilitated diffusion, certain hydrophobic molecules such as glucose, amino acids, and ions are transported passively down their concentration gradient by carrier-mediated facilitated diffusion or channel-mediated facilitated diffusion. So essentially what this is doing is that it's essentially operating the same way. You're going from higher concentration to lower concentration, but in this case, you're using proteins to do that. Or a channel. So it could either be a carrier, which is the prior one, or just a channel, which is sort of just like an open tube. So osmosis, this is the movement of a solvent, not molecules, such as water, across a selectively permeable membrane. Water diffuses across the membranes through lipid bilayer. Even though water is polar, it is so small that some molecules can sneak past through specific water channels called aquaporins. The flow occurs when water or other solvent concentration is different on the two sides of a membrane. So this is where we can set up what's called an osmotic pressure, where you have, let me go to an example here. So here we look at this example and you're taking red blood cells and you're putting them into either water, salt water, or what we call an isotonic solution. So one thing I want you guys to realize is inside that red blood cell, you have solids, you have proteins, you have carbohydrates, you have salts that are all found inside, right? So if you put it into an isotonic solution, which means solute-wise it's equal to the inside of the cell, you're going to see nothing happen. But if you put it into hypertonic solution, which would be something like salt water, meaning now you have more solids outside than you have inside the cell, what will happen is the water inside the cell will actually leave the cell and the cell will shrivel up. In example C here, a hypotonic solution, what's happening is you're putting it in just pretty much plain old water and there are no solids in the water. So what happens is because you have all those solids inside the cell, the water is going to rush into the cell to try to balance out, to try and equal out. And what's going to happen is the cell will swell, and at some point it may even explode. The final type of transport is active membrane transport, and there's two types. We have active transport and vesicular transport. Active transport is defined by requiring ATP to move solids across a plasma membrane, either because the solid is too large for channels, the solid is not lipid soluble, and it is not able to move down concentration gradients. Active transport will require carrier proteins. And we have what's referred to as primary active transport, where the required energy comes directly from ATP hydrolysis. Secondary active transport required energy is obtained indirectly from an ionic gradient created by primary active transport. What that means is you're expelling the energy to move one thing and something else is sort of piggybacking onto that movement. So primary active transport, the energy from the hydrolysis ATP causes a change in the transport protein. The shape change causes solids bound to the protein to be pumped across the membrane. Example would be sodium potassium pump. And you can read about that here. So the sodium potassium pump, this is the most studied pump. It is an enzyme called the sodium potassium ATPase that pumps sodium out of the cell and potassium back into the cell. It's located in all plasma membranes, especially active and excitable membranes such as nerve and muscle. So you have leakage channels located in the membranes that result in leaking of sodium into the cell and leaking of potassium out of the cell. Both are traveling down their concentration gradients. But because of the sodium potassium pump, this is going to pump the sodium back out of the cell and the potassium back into the cell against their concentration gradients. That's why it requires energy, because you're pumping the stuff back in against concentration gradients. This helps to maintain electrochemical gradients, which involve both concentration and electrical charge of ions. And this is essential for muscle and nerve tissues. And we'll talk more about this when we get to muscle physiology. That's where we'll see it first. We'll also see it with nervous physiology. Secondary active transport, this depends on ion gradient that was created by primary active transport. So energy is stored in gradients, is used indirectly to drive transport of other solids. Low sodium concentration that is maintained inside a cell by the sodium-potassium pump strengthens sodium's drive to want to enter the cell. Sodium can drag other molecules with it as it flows into the cell through the carrier protein. So here we have our primary active transport, as you can see here in the dark purple, and our secondary active transport as a result in the light purple. Vesicular transport, this involves the transport of large molecules, macromolecules, and fluids across a membrane in a membranous sacs called vesicles, which usually requires cellular ATP energy. Vesicular transport includes processes such as endocytosis and exocytosis. So essentially, you're moving things into and out of the cell by vesicles, membrane vesicles. Endocytosis is transport into the cell. Exocytosis is transport out of the cell. And then you can have other types based on endocytosis and exocytosis. So here you can see this is endocytosis. You're coming in through a vesicle, okay? And then you can see you can also exit through a vesicle. White blood cells will use a type of endocytosis called phagocytosis. It is an endocytic pathway where you might get the impression of cell eating, okay? We'll talk more about this when we get to the immune system with phagocytosis. Pinocytosis, this is sort of bringing fluids in through vesicles, and this is what we refer to as cell drinking. You also have receptor-mediated endocytosis, which means endocytosis only occurs if a particular substance binds to a receptor. This is something that we'll talk about when we get to the immune system. And phagocytosis is essentially using receptor-mediated endocytosis as well. With the process of exocytosis, here you can see you have the membrane-bound vesicle migrates to the plasma membrane. The proteins will interact with each other, and then the vesicle and the plasma membrane will fuse. The vesicle contents then are released to the exterior. Okay, we're about halfway through the presentation, and I'm going to end this recording here and pick up with a part 2 recording.